Bacteria are prokaryotic microorganisms which play a vital role in the life of human beings. Alexander Fleming first found out that bacteria can be used in field of medicine. What he did was he was experimenting on Staphylococcus aureus and he was culturing the bacterium in a petri dish. What is left was a orange which got rotten and was close to the petri dish. There was something which was secreted from the rotten orange that caused the Staphylococcus aureus colony to get degraded. If you see this culture, you will see that these all dotted lines or these all dots which I have colored green, they all are the colonies of Staphylococcus bacterium. But the white color part is the portion where the orange secreted or the rotten oranges, some um, fluid came into contact with the culture, and because of that there is no colony of staphylococcus present nearby. This made Alexander Fleming conclude that there is something secreted by the rotten oranges colony or that blue colored colony which has caused the staphylococcus bacterium to lyse its outer cell wall or cause its death. And this was the very first time penicillin was invented from Penicillium notatum. The blue colored fungal growth on the orange, rotten orange, is because of penicillium and it secretes penicillin, a chemical which is capable of killing the or dismantling the outer cell wall of the Staphylococcus aureus. If we so let's look up to the various uses of bacteria in the field of medicine. First is the antibiotics. Antibiotics are the chemicals or substances which are released by the bacterium in order to slow down the growth of bacteria or any foreign antigen. Now the example is of same Penicillium notatum or Penicillium chrysogenum is capable of secreting a chemical, and the name of the chemical is penicillin. This penicillin is used in order to treat the individuals who have some bacterial attack. This is the culture of bacterium growing over a agar plate. Serum is a clear straw colored liquid which is separated from the blood, and it is rich in salt, proteins, glucose, and fats, and it also contains antitoxins. These antitoxins can be used in order to treat some other patients who are suffering from some disease. Now, you should be aware about the difference between serum and plasma because it is also separated from the blood. When there is a blood clot formation taking place, whenever there is a cut in our body, we see that there is presence of some yellow fluid which is secreted other than the blood clot. That yellow fluid is the serum, and we can use it because it has presence of some it can be given to any individual who requires immunity. Antibodies are substances which are formed in our own body against any foreign substance which has entered. The substance which enters from outside can be a bacterium, a virus, or a parasite, and it is known as an antigen. Whereas antibodies are released in response of antigen in order to kill the antigens or make them non-functional. If we see this structure, you can very well understand it that this Y-shaped structure which is visible is the antibody, whereas this is the bacterium and it is the antigen. And what is the function? Every time one antibody will go and bind with the antigen and kill the antigen. And because of this, the individual, if anyone is suffering from the disease, they will be cured. Antivenom is made by collecting venom from the animal, and what exactly is done is the venom is taken and it is injected in the animal. After injecting the venom in any animal, the animal starts secreting antivenom, and that antivenom can be taken out from the blood serum and can be given directly to any individual who has suffered a snake bite, or has suffered from scorpion bite, or any infectious venom attack. And the action of this antivenom is way much rapid than the WBCs forming the antivenom. Some of the animals, they are immune to the attack of snakes, and that's why we can harvest the antivenom from them. A very common example is of a horse. So what is done is the poison of the snake is taken and it is injected in the animal, which is a horse. From the horse's blood, that blood is taken up from the horse's blood after a certain period of time, so that the antivenom is already present in the blood. From that blood, plasma is separated, and that plasma is used to act as an antibody, and it is stored in a formulated form, because serum cannot be given directly to an individual. We need to make it usable, and when we make it or we extract the usable form of it, we use it in form of an injection in order to treat the venom attack. And as because most of the venom attacks, they are neurotoxic, so we need very quick response, or we need very quick medication. That quick medication can only be given by the synthetic or already prepared antivenom. Antiviral drugs, these are the medicines which are capable of destroying the microbes or inhibiting their replication. Replication means duplication of these viruses. So, whenever there is a viral attack, we take some antiviral drugs, and the function is to just reduce the viral load. It means that the number of viruses which have infected us, suppose 5 viruses has entered one individual. Now when we want to decrease the viral load, it means that we want to stop the viruses from becoming 10. From 5 we will not allow them to become 10 or 15. We will let them be only 5, and our body's immune system will get activated to release the antibodies. The antibodies will come and kill or neutralize the effect of the viruses which have entered our body. Antidotes are agents that negate the effect of poison or toxin. Antidotes mediate its effect either by preventing the absorption of the toxin, by binding and neutralizing to the poison, or antagonizing its end organ effect, or by inhibition of conversion of the toxin to more toxic metabolites. It just attaches Antidote just attaches itself, or um, think about a chemical which is as such that it will just go and attach with the toxin which is released by the bacterium. And when the toxin is attached with some antidote, its pathogenic nature or the disease causing nature will be depressed or will be suppressed. And because of that, and how does it happen? It happens because of neutralization reaction which is taking place between the organism as well as the toxin. Vaccination is the administration of vaccine to help the immune system develop protection from a disease. So before we suffer from a disease, we take or we form the antibodies with the help of weakened germs or dead germs, or just introducing the toxins type of substances within our body, so that our immune system is already aware how to denature the effect of the bacterium. The exact way how it works diagram where you can see a cell. This cell is as such that here this is the bacterium which has entered, and as soon as the bacterium comes in contact with the T helper cells, the T helper cells combine with the antigen, and the antigen presenting cell along with the T helper cell kill the virus which has attacked the antigen presenting cells, and as a result there will be neutralization reaction and no problem will be there. There will be no visible symptoms of the viral attack. There are two things which take place during this process. One is the killing of the virus which has taken place or the bacterium has taken place, and second thing is the formation of memory cells. So next time when the similar kind of bacterium enters our body is capable of fighting against it immediately. So stored information for killing the entire virus or bacterium is recorded in our body in form of memory cells. The types of vaccines are there are various types of vaccines. First is live attenuated vaccine, tuberculosis, oral polio vaccine, measles, rotavirus, or yellow fever virus. Now, as the name is live attenuated, second type is inactivated or killed antigen. We just kill the antigen and just the outer cell wall of the bacterium is used in order to form the antibodies in our body, and then our body remembers the technique of killing the bacterium. Subunit purified antigen, where just the antigens are used, and toxoids are also there, which are inactivated toxoids which can be taken up in the form of vaccines. Live attenuated vaccines, how are they formed? uh, See, in basically you can think that the virus is introduced to a culture of any animal. A virus when it is infecting a human being, human beings are developing the symptoms, but the same virus when it affects the animals, it does not show the symptoms in that to that extent. So what we do is we introduce the same bacterium which is about to cause disease to us into a chick, and into a chick embryo, and now we cultivate the or we allow the bacterium to grow in the same chick embryo for 200 generations. So 200 times the chick is giving rise to next chick, then again next chick, from first chick to second chick to third chick, till 200 times. So as the bacterium is growing in the same chick, it becomes attenuated, the chick becomes attenuated to the virus. It is the virus is now no longer capable of transmitting any disease to either the chick, nor to neither the chick nor to the human beings. So now that virus which has been growing for 200 generations in the chick can be taken and can be introduced into the human being. Since that virus is growing in the chick for several generations, now the pathogenic nature of the virus has decreased. Now it is no more equally pathogenic, so it will not cause disease or much harm to the host or to any individual to whom it is given. And as a result, the individual who is taking the virus will develop the antibody for the same virus, but they will not get infected by the virus. This is used in order to this kind of technique is used to form the tuberculosis vaccine, which is BCG. Bacilli calmette urine is to be learned. You can see the entire chart where different viruses are getting killed, or we are forming virus vaccines with different techniques. First technique is live attenuated virus, which we have already discussed. If a particular virus is grown for 200 generations in any other animal, and then after the same virus is taken up or is introduced to any human being, then the capability of causing disease from that bacteria is decreased or it is completely lost. Virus like particles, similar structure of the virus, that kind of particle can be introduced in the body of any human being, and as a result antibodies will be formed. But our body will no longer, or our body will not be suffering from any disease because of it. So that is an idea. Third idea is recombinant bacteria. So we can just make a bacteria in such a way which can kill the virus. That can be done. DNA carriers can be used, or recombinant viral vector can be used. Recombinant subunit viral portions can be used. Just the outer receptors can be studied, and with the help of that, we can make a bacterium which is incapable of causing any disease. Synthetic peptide, just the capsid structure of the virus can be taken and it can be used in order to form the vaccine. And whole inactivated virus, the entire virus after killing, can be introduced in the individual just for the process of immunization. Killed or inactivated vaccine. So one alternative to attenuated vaccine is killed or inactivated vaccine. We take the same virus, but in spite of attenuated virus from the chick embryos of 200 generation, the last generation, we take the bacterium, and in spite of introducing in a live condition to the human being, we kill the virus or the bacterium by heat or by formaldehyde or by some chemical, introducing it to some chemical. Now the dead virus or bacterium is introduced into the individual who wants to gain immunity against it. That is done in case of polio or hepatitis virus. So that is one technique by which we can treat the individuals, by heat killing it. Here also the same technique is given. um, you can see the inactivated virus. That is what we did studied now. Toxoids are certain toxins or chemicals which are secreted by the bacterium when they are introduced to stress situation. So the symptoms are as such, like one example is there for tetanus. Tetanus is a disease in which jaw locking takes place, or it is a neuro um, paralytic disease. The nervous system is paralyzed because of the attack of Clostridium tetani, a bacterium. So, immunization for this type of pathogen can be made by inactivating the toxin that causes disease symptoms. As with organisms or viruses used in killed or inactivated vaccine, this can be done via treatment with a chemical such as formalin. So, we are just using certain chemicals in order to kill the Clostridium tetani and then introducing it into any individual. That is what we do when we take the vaccine for, or when we take the injection for tetanus. So this is another technique. So you can just look into this diagram and you can very well understand that diphtheria bacterium emitting toxin, that globular structures are the toxins which are secreted by the rod shaped bacterium, which is diphtheria. And after that, that toxin is taken and it is modified in such a way that you can see that red color something is attached to the toxin. Now that thing will be introduced to the individual who is suspecting to be suffering from the tetanus or is interested in taking vaccine against the disease. So this form which has red color substance over it is chemically inactive and is harmless, but when it is introduced in the individual, the body learns how to kill the toxin released by this tetanus bacterium, and as a result the individuals, they become attenuated and they do not suffer from the disease at all. This is the economic importance of bacteria.